Hi, my name's Art Krieg, and I'll be talking about retroviral mimics and their potential role for cancer immunotherapy. My disclosure information, in particular the presentation I'll be giving, is in my capacity as the founder and CEO of a new biotech company, Zola Therapeutics, which is developing these compounds. Now, what I want to talk about today is the idea of inducing endogenous anti-tumor CD8 T cells in situ to fight cancer. And of course, we know that CD8 TILs are associated with survival in all different types of tumors. When these T cells are present, they're able to attack the tumor, especially if you unleash them by blocking the checkpoints that can hold them back. But of course, most patients have inadequate TILs present, and so that brings us back to the idea of inducing these in patients where there aren't enough or where they're inadequate. And the way I want to approach this conceptually is to start from the self-evident fact that CD8 positive T cells have evolved for the purpose of killing viral and retroviral infected cells. And so if we can mimic the signals that the innate immune system uses to distinguish that type of infection from self, then we should be able to induce those cells at will to fight cancer. Now, we know that retroviral nucleic acids can only be distinguished from self via three different receptors. Toll-like receptors 7 and 8 distinguish uridine in viral RNA versus pseudouridine in the most common self RNAs. And these receptors are expressed in virtually all of the dendritic cell populations and myeloid cells. I'm abbreviating them macrophages here. And then TLR9 detects unmethylated CpG dinucleotides in viral DNA, whereas in our self-endogenous DNA, the uridine in endogenous self DNA, the cytosine is generally methylated to 5-methylcytosine. And TLR9 is much more narrowly expressed in a subset of dendritic cells, plasmacytoid dendritic cells, and in B cells. Now, we already have published data that a TLR9 agonist, a CpG DNA, can induce tumor regression. Vidutolimod, Giorgio Trincheri yesterday talked about the intratumoral treatment in neoadjuvant melanoma, where very nice responses were seen. We've also seen monotherapy responses in advanced PD-1 refractory melanoma. This induces TILs. It can induce systemic regression, including non-injected metastases, as I'll come back to. But what I want to leave you with today is that we can advance from the positive results with just stimulating TLR9 if we add the stimulation of RNA through Toll-like receptors 7 and 8 in the right way. We can mimic the entire retroviral replicative complex in a way that evolution detects, and this should induce higher anti-tumor CD8 positive T cells. So that's the goal for today. Now, you won't find any of this in the cancer immunity cycle. That's great as a high-level overview, but there are three key concepts that I think are not adequately explained in standard review articles of cancer immunity. One is the difference between different TLRs. You won't find anything about TLR7, 8, 9 in the cancer immunity cycle. Differences between different types of dendritic cells, plasmacytoid dendritic cells versus others. And fundamentally, the fact that type 1 interferons themselves are very heterogeneous in their function. And I want to start by talking about the difference between interferon alpha and interferon beta. Now, there are 13 different genes for interferon alpha. These are non-redundant. And so you have to ask yourself, why would 13 different genes have been conserved through human evolution, whereas for interferon beta, we just have a single gene? These, of course, all bind to the same receptor, the IFNR heterodimeric receptor. The affinity of interferon alpha is 1/10 to 1/100th the affinity of interferon beta. So when both of these are being produced, interferon beta is going to dominate. Now, the biologic effects of these in humans are very different. In the 1980s, when recombinant interferon alpha and beta were first produced, they were tried in all kinds of different clinical indications. And very quickly, it became apparent that interferon alpha could occasionally induce regressions in cancer and some chronic viral infections. And it's been approved for those. It actually makes multiple sclerosis worse. Interferon beta, on the other hand, never achieved an approval for chronic viral infections or cancer, despite many clinical trials over more than a decade. Where interferon beta got approved was in the treatment of multiple sclerosis. And it's been shown that it induces regression in multiple sclerosis by activating Tregs and by inducing IL-10 secretion. So interferon alpha and interferon beta in humans are fundamentally and biologically different. And I think one point that is often missed in our field is that in mice, this difference doesn't really exist in the same way. In mice, interferon beta was discovered again in the 1980s and 1990s, where it was studied. It induces CD8 T cells against tumors perfectly well. In fact, in some models, it's superior to interferon alpha. So for that reason, mouse models where type 1 interferons are involved do not predict human efficacy. And so the proposal that I want to have, and which is underlying my presentation, is that for human cancer immunotherapy, the goal is not just to induce type 1 interferons. The goal should be to induce the highest possible ratio of interferon alpha to interferon beta to take advantage of this biology without this biology. So in order to do that, the cell type that we need to activate is the P1 plasmacytoid dendritic cell. More than 90% of the interferon alpha that's produced in response to infection comes from that cell. And there are only two receptors that are expressed in the PDC that can induce strong interferon alpha production, and those are TLR7 and TLR9. In contrast, interferon beta can be produced by essentially all nucleated cells, and essentially any of the pattern recognition receptors can induce interferon beta to varying degrees. So let's talk more about the P1 PDC. And this is all published work, but it's not work that has been widely recognized in the cancer immunotherapy field. Resting PDCs are negative for PD-L1 and CD80. And when they're stimulated for 24 hours with flu virus, this is a strong stimulus, they differentiate into three distinct subsets. And those are transcriptionally distinct, but what's really important is their functional distinction. The P1 subset makes interferon alpha, and this subset has been reported to be important in the defense against COVID. In contrast, the P3 subset actually promotes Th2-like responses, and there have been reports that the P1 and P3 subsets are inversely associated with melanoma survival. So for cancer immunotherapy, you want high P1s and you don't want P3s. So how do we induce P1 PDCs? In the same manuscript, the authors looked at a range of different innate immune activators, including all three of the major types of CpG oligonucleotides. And what I've done is to extract the data from their paper into this table, where I've ranked these various agents by the percentage of P1 PDCs that they induce in normal purified PDCs. And then I've also calculated the ratio of P1 to P3 PDCs, if we think about this as a kind of rheostat that's adjustable. So among published agents, the CpG-A induces the strongest P1 to P3 ratio. And in the clinical trials, as I mentioned before with vidutolimod, a CpG-A DNA has shown monotherapy activity. In contrast, agonists representing these other classes have been in human clinical trials for cancer immunotherapy as well. Tvec, of course, is an approved oncolytic virus. It's not exactly the same as flu virus, but Tvec is more known for activity locally with less evidence for systemic tumor regression. CpG-C oligos, this is tilsatolimod and SD101, those have not shown monotherapy activity in cancer. Likewise, small molecule agonists related to R848 have shown occasional responses, but not that impressive. And the CpG-B oligos also very rarely responsive. So among these, only the CpG-A has been shown in humans to induce more than a 10% response rate. And so I want to just briefly touch on how CpG-A differs from the other types of CpGs. And I show here the sequences. These all have been in human clinical trials. The CpG-B was the first, starting human clinical development in 1999. And at that time, the assay that we had for activity was B-cell activation. PDCs hadn't even been discovered yet. Promune failed in phase 3 clinical trials of cancer, but CpG-B oligos are approved as vaccine adjuvants. SD101 and tilsatolimod are similar in having this phosphorothioate backbone to make it nuclease resistant, but again, that has not shown strong activity. What has is the native phosphodiester backbone that is in vidutolimod, and it turns out that for P1 PDC activation, there's an obligatory cleavage step by DNase 2, and you need a phosphodiester DNA backbone to have that in order to get the maximal P1 PDC induction. These G regions on the end form G quadruplex structures, and these are present in the genomes of viruses and retroviruses that infect humans. So mimicking the retroviral DNA genomic structure is associated with efficacy in advanced melanoma therapy. This is what vidutolimod looks like. The CpG-A cargo is packaged inside a Q-beta bacteriophage virus-like particle, and intratumoral injection of this induces TILs in the injected lesion and also in non-injected lesions. And yesterday, Giorgio Trincheri presented some of the neoadjuvant data from Divakar Davar's study, where the major pathologic response rate was 57%. And this efficacy was comparable to the Ipi-Nivo data reported in Christian Blank's recent New England Journal paper. But the safety of vidutolimod plus Nivo appears to be superior in terms of grade 3 treatment-related AE, 21% for Vidu versus almost double that with Ipi-Nivo. As a monotherapy in PD-1 refractory advanced melanoma, 9 out of 40 responders, and these included systemic responses in 4 patients who happened to have systemic lesions that could be measured, including liver mets in 1 patient. And 2 of these responders previously had progressive disease in a different trial with TVEC plus PD-1. So vidutolimod may be biologically different from oncolytic viruses, or at least from TVEC. And then in advanced melanoma, similar response rate, but longer duration of response. And again, abscopal effects with systemic regression. These responses were associated with increased CD8 TILs, CD4s, PDCs, tertiary lymphoid structures, and macrophage activation signatures. So we know what to look for as we go forward with development. And just to give an idea of the magnitude of some of these responses, this was a patient who had been heavily pre-treated, had a melanoma groin lesion, a lymph node that was injected, and just with injection of that groin lymph node had systemic regression of these large lung mets with a long-term survival. Now, we do know there are limitations in the activity and the responses to vidutolimod, and one of the major issues is the immunogenicity of the virus-like particle. If you look at antibodies to the VLP, they're rapidly induced in all patients. And those antibodies, if you add a monoclonal anti-VLP antibody in human PBMCs, and you just measure interferon alpha production, which is a surrogate for P1 PDCs, you can see that as you go up in the antibody concentration, you lose that P1 PDC induction. And that's mediated by myeloid cells because we don't see that regression in purified PDCs. So the conclusion is that this VLP delivery, by being immunogenic, this is apparently enabling myeloid suppression of the P1 PDCs. So to get around that, we have decided to change our delivery of a CpG-A to a lipid nanoparticle, which still will activate the PDCs, but without the production of neutralizing antibodies, and we can optimize LNPs for uptake into immune cells. Now, if we're only activating the PDCs, though, we're missing out on the opportunity to harness CDCs and TAMs to also contribute to T cell activation. And so what we'd like to do then is to add a TLR7/8 agonist to co-stimulate the PDC, but also activate those other cells. And of course, conceptually, that would then mimic a complete retrovirus and potentially harness the evolutionary drive to generate T cells more effectively. But what TLR7/8 ligand should we use? Because there's actually two different major types of TLR7 agonists. Most clinical development to this point has used small molecules that bind the TLR7/8 homodimer at a site that's been called the first site. And if you look at the biology of these small molecules in human PBMCs, they induce very high levels of IL-6 and TNF-alpha production. And that's because the first site induces the NF-kappa B pathway. It does not particularly induce the IRF pathway that leads to interferon alpha. To do that, you need an RNA agonist to bind also the second site in the homodimer. And so if you have an optimized RNA agonist, you can induce strong interferon alpha production, but you actually have decreased the NF-kappa B activation. And so what I would submit to you is that for cancer immunotherapy, we don't want this type of profile. We want this type of profile to get high interferon with low inflammatory cytokines. Now, how does this compare to TLR9 agonists? In the past, the CpG-A DNA that I showed before was the strongest interferon alpha inducer that was published. These optimized agonists that we have now developed are superior to that. And a key feature of these agonists is that we are using phosphodiester RNA now. And that, again, allows nuclease cleavage. So we get a stronger induction than we achieved with the TLR9 agonists. In the past, with the RNA agonists that we had been developing, we did not see synergy when we would mix those with the DNA. We now see a very strong, more than additive stimulation of interferon alpha with combining a TLR7/8 RNA with the DNA. And we see this without strong induction of the inflammatory cytokines. Now, it's important to note that TLR7 and 8 are on the X chromosome, and there have been reports that they may at least partially escape X inactivation, and that females respond more than males to other published TLR7/8 agonists. So we wanted to look at this with our optimized RNA agonists, and as a first step, I'm just going to show the data for the first two male and female responders in, again, normal PBMC assay. I have a legend on the bottom here to show you R848 for each of these panels is on the right. The TLR7/8 RNA data are on the left, and then the TLR9 CpG-A, and then the combination of the two. And so for inflammatory cytokines, R848 again is the highest, and there's a trend that women have higher responses than men. For the interferon alpha response, again, R48 is very low. Women do seem to have a higher response to the combination, whereas with the TLR9 agonist alone, it's the same between the sexes as would be expected. So based on this, we have decided to move forward with clinical development of separate TLR7/8 and 9 agonists so that if we need to adjust the doses based on sex, we can fine-tune this. But it's definitely something we have to be aware of in clinical development. So our plan then is to develop 2 products, not just one, to stimulate these receptors. ZU09 to activate TLR9, ZU07, the RNA, to activate these other 3 cell populations. Together, these are the cells that are required to maximally stimulate CD8 positive T cells. They will still have checkpoints on their surface, and so ultimately we're expecting combination therapy. But by activating these receptors together, we are mimicking a complete retrovirus, and this should provide greater induction of the CD8 T cell responses. So where do we want to develop this? We would like to see if we can activate liver TAMs in liver metastases. And of course, we know from the great work of Yu et al. In Nature Medicine a few years ago that these TAMs suppress CD8 T cell responses, and that may be involved with the worst outcomes in patients who have liver metastases. CRC is the most common source of liver mets, and so this could be an area for us to start. We did try intratumoral injection of vidutolimod in patients with MSS-CRC liver mets. That didn't work, and this was done at Sheba. We did not see clinical response. We also did not see any of the response biomarkers. So activating TLR9 alone in those liver mets isn't enough. Can we develop an IV agonist to stimulate all 3 of those receptors to convert MSS-CRC liver mets into M1-like state? I'm just going to show the mouse data where we did test IV vidutolimod in mouse orthotopic liver cancer models. We saw anti-tumor activity in the mice with CMP-001. This is tumor growth in the controls. This is the sorafenib standard of care, and the CpG was similar to sorafenib. In a second orthotopic liver cancer model, we again, and in this model, anti-PD-1 did not have anti-tumor activity compared to untreated controls. IV vidutolimod inhibited tumor growth. Subcutaneous vidutolimod inhibited less, and then this is just a no tumor control. So why would IV vidu work in mice, but intratumoral vidu doesn't work in humans? It could be the difference in TLR9 expression. In mice, TLR9 is expressed in all of these cell populations, so they will all be activated by vidu. In humans, TLR9 is not expressed in the myeloid cell populations, so we're not activating TAMs. So for TLR7/8 agonist, if we deliver this into the liver TAMs, we may be able to make this like the mouse response to vidutolimod. And so lipid nanoparticles are the way that we're thinking about delivering this. And that's based on a lot of human experience with IV LNPs, with other nucleic acid therapeutics, siRNAs, gene editors, base editors, and so forth, all use lipid nanoparticles to deliver into the liver. There's rapid uptake after IV injection, and although most of these compounds are targeted to hepatocytes, the highest level of uptake actually is in Kupffer cells and other immune cells. And of course, these are the cells that we're interested in targeting in cancer immunotherapy. So how are these cells taking up the LNPs? There may be multiple mechanisms, but one of these may relate to CD36, which on the first day of this meeting, Sue Keck mentioned is upregulated in tumors. It's a scavenger receptor involved in lipid uptake, and this is a paper from the Dicerna group, now at Novo, showing that in tumor-draining lymph nodes in a mouse model, there's higher expression of CD36 compared to normal lymph nodes. And you can take advantage of that using a lipid-conjugated siRNA that can target CD36. You can achieve knockdown of a target using siRNA in tumor-draining lymph nodes without knocking the target down in normal lymph nodes. So it turns out that if we look in data from single-cell RNA-seq from pancreatic cancers, both in the pancreatic cancer itself and in liver, that the CD36 expression plotted on the y-axis is correlated with CD163 as a marker of M2 cells. So it may be possible to target M2 cells in tumors if you optimize your lipid lipid nanoparticles for delivery to CD36. And interestingly enough, an M1-like marker, CD8 is actually not expressed with this. And this is thanks to our collaborators at the Broad Institute, Sarah Kong and Arnav Mehta. So we have developed a couple of different formulations. This is just showing our very first monkey data, which we just got in the last few days, using an IV formulation of a pretty standard LNP using the lipid MC3. And again, the cargo here is an optimized phosphodiester backbone RNA to activate TLR7 and TLR8. There are 6 monkeys in the study, and we did IV dosing with weekly dosing at increasing dose levels from 20 micrograms up to 2 milligrams. Most of the data are still pending, but I'll just show data down here, CD86 expression in the myeloid dendritic cell population at baseline, week -6, week 2 pre-dose, week 2 post-dose in both males and females, seeing an induction there. We don't yet have the week 3 data for the males to know with this formulation, but at week 3 with the female, we see again that same increase. Much more data to come there, and we're moving forward also with mouse models, of course, and looking in human tumor-associated cells. But what I'd like to turn to next at a high level is what we think may be going on. I think it's widely accepted in the field that pattern recognition receptors evolved to detect different types of danger. And at a high level, there's infectious danger and there's non-infectious danger due to tissue damage. And it makes perfect sense that different combinations of pattern recognition receptors have evolved to distinguish these types of danger because you need to make different responses. You need to induce CD8 T cells to kill retroviral infected cells. You don't want to induce CD8 T cells in the setting of sterile tissue damage. And so tumors mimic damaged tissue. They activate the cGAS sting pathway, which appears to have evolved to detect cell stress. This does not distinguish self and non-self. And in humans, there are data suggesting that that can contribute to tumor progression. On the other hand, TLR7, 8, and 9 clearly do have the potential to distinguish retrovirus nucleic acids and viral nucleic acids from self. And so it makes perfect sense to stimulate those receptors if you really want to induce a CD8 T cell response. And of course, we also have the evidence with vidutolimod that even just activating this receptor alone can have some activity. So I think that activation of these 3 receptors in combination should induce optimal CD8 T cells, not to say you couldn't combine it with other agents. And so in conclusion, building on the positive data with vidutolimod, we'd like to combine that with an optimized TLR7/8 agonist, and we'd like to try to develop this for systemic delivery. And we're thinking about clinical evaluation of this approach through the IV route in patients with resectable liver metastases for initial human investigation in a window of opportunity study. Can we convert TAMs into M1-like activated myeloid cells? And that should be a relatively quick readout in such a study. Intratumoral injection, obviously for neoadjuvant therapy especially, is very appealing. This could be used with vaccines as well. It should be the strongest adjuvant to drive CD8 positive T cells. And then, of course, other routes of administration are also possible. So finally, I just want to acknowledge the Zola Therapeutics team, our collaborators at the Broad, our CROs. We don't have our own labs, and so we're dependent on our CROs. The oncology advisors, many of whom were involved in the clinical development of vidutolimod and other CpG oligos. And then finally, the patients in prior CpG trials. Going all the way back to 1891, we named Zola Therapeutics in honor of Zola, the first patient treated with live bacteria, actually, by William B. Coley. And there's a marvelous description of Zola's experience. And we hope to bring improved cancer immunotherapies to patients. And with that, I thank you.